Hi, welcome to Art of Academia, a weekly podcast featuring interviews with leading scientists and insider takes on life in academia, hosted by Komal and Madan, researchers from Cancer Science Institute, Singapore.
Okay, do you want to start with your Thailand trip? Just came back. What was that like? Third time.
Thailand feels like home now. Seriously, I was just telling my friend, Thailand just feels like home. Good fun. Went to Krabi this time. Near, very similar to Fifi. Like all of it is very similar.
Yeah.
They're very close by. The
Yeah.
ocean is the same. This green, like pretty beautiful place.
Does it have the streets like Fifi? PP, sorry.
Oh yeah, much smaller.
Okay.
So like there was just one walking street which had similar vibe as Fifi Market.
Does it have like the same tiles and stuff? I'm just curious.
No, it does not. Yeah, but the vibes are very similar because the things that they're selling and the people that there are, the food,
Fair.
all of that is very similar. Definitely, I still love Fifi. I will go there again.
Again?
I will.
Okay.
I'm not kidding. Yeah.
Okay. Take me next time.
I will. Maybe next year, not this year. I can't do more this year. Oh, we did a little hiking where it was like a hill. So we are doing it. This other girl is in her slippers, and I'm like in my shoes, luckily. There's a snake coming,
Oh.
like crossing the way.
Like coming towards you?
No, like
It's
crossing
just
the way right in front
across.
of. I'm standing here. The snake is right there. You're like, are we gonna go back? But then you're like, no, let's go.
Even with the slippers on.
Yeah,
Brave.
yeah. That was good fun.
Do you know how to tell if a snake is poisonous or not?
I think if they are brightly colored, they're
Is that true for
poisonous.
snakes?
I don't know. That's
I
more
think
like
that's
frogs.
like frogs. Yeah. And maybe mushrooms. I think that's true for snakes.
Oh, we saw the plankton this time.
Oh, the luminescent
Yeah,
one.
that was nice. That was
Like,
great.
what were you were on the beach or
No, so this is part of the trip.
on the boat stuff?
Yeah, so the boat comes very close to the shore. You get down, and then you try to see under the boat because it has to be dark. And then you move your hand, and then you see them sort of. I think when they move, they bioluminescence or something like that.
Yeah, I don't know how they work.
Yeah, but that
Never
was
seen.
fun.
So the thing is, when we went to Bintan, we were all like swimming, and then once we came back, Juan was like, "Yeah, there was bioluminescent plankton." I'm like, "What do you mean there was?" He's like, "You were swimming in it." I was like, "What?" I don't remember. I didn't notice. No one
Really?
told me. Yeah, so apparently it's common in Bintan, and they have
Okay.
seen it multiple times.
Oh, crazy!
I was there 3 times. I never saw it. And he's like, "No, you saw it." I'm like, I don't know. I saw it.
How was your weekend? How was the game night and stuff?
One of our friends, she introduced us to the Dungeons and Dragons thing.
Yeah.
We didn't play it.
Yeah.
So you have to first create your characters on there's like a website. And there's like 100 things you have to decide. Apparently.
Oh my god, I did not know that.
And then
So this character just depends on me, however I want them
Yeah,
to be.
you can
There's
do-
no restriction.
I mean, so the backstory and all is like free. You can do whatever. But I guess there are some guidelines. And there are restrictions on, I guess, attributes. Right? You can't
Yeah.
make everything
Yeah,
full.
I'm
no?
invincible.
And there are like classes also.
Okay.
So there are like different types of people. So
Uh-huh.
paladin, warrior, druid, whatever. So they
What's a
come.
druid?
Druid is shape shifting. People who can shift into animals.
Damn, I'm so excited.
Yeah. So it's very complicated, but
Yeah.
quite interesting.
Okay.
We'll try. And they will also have props. So she said it'll be
Nice.
easier to like imagine the
Okay.
scenario.
I'm too excited right now.
Yeah, so we'll do that. I haven't started. We'll get to it.
Okay.
Okay, so we said we'll do. Okay, so this is very inspired from the Not Overthinking podcast because they kind of do this.
Yeah.
So they usually start with like what's going on with life. So
Yeah.
this includes things like where have you been traveling, what have you
Yeah.
been up to.
Yeah.
Like I'm struggling with this thing, right? So very funny for Taimur, the brother. One consistent problem is like he struggles to poop in the morning. Like he just spends too long on the toilet, and that annoys him.
Okay.
Right, so occasionally this comes up, and then usually, and this has become a meme now. So whenever Ali talks house life, he'll be like, "Been pooping well" or whatever. But something about it is like,
Boy.
yeah. So he's like talking through his thing of like, this is his problem, right? What like why
things
it
he- what
annoys
is he
him
trying
and
to
what he
do?
wants to do about it.
Yeah.
Even though it's something so silly,
Okay.
I think that it's gripping for some reason. And
Right.
sometimes it's more real, tangible stuff, right? Like sometimes it's like, oh, my calendar is too full these days. Like I want to
Sure. I mean,
wanna
that's.
break it down.
Yeah.
Okay, so let's do that. So I was thinking, what have I been up to last week, right?
Yeah.
And yeah, so I've been thinking about switching to Quarto. Have I mentioned Quarto to you?
I think you might have, but go on.
Okay, so this is basically, okay, I don't want to get into too much of the details. So you know how when you do your analysis, there are sort of notebooks that you can do them on. So Python has IPython notebooks, which is very popular. And then R used to have like it was just R Markdown before. So what the concept of these is that you run your analysis from a like you write your code on a document, and then when you press run, it runs and it shows you the result below it. And when you save it, it saves it into one document.
Oh yeah, you.
So that when you open it up later, you see the code and you also see the computation.
Right there, yeah.
Right, so that's like the basic thing. And Quarto is like sort of this new and improved way of doing this that has other advantages. I won't go into it, but it works with both R, Python, whatever.
Okay.
But the main thing is like I don't use them. I don't use notebooks in my workflow. I have scripts, or sometimes I'm just writing things directly into the terminal.
Yeah.
But I try to document them.
Yeah.
Like I spend quite a lot of time on documentation. And the reason I never went to notebooks is because on some hand they feel a bit clunky. Like to me the script just feels like more smooth, it's faster. Right? You feel that with terminal also. Instead of like opening up a file and writing your code in and then running it, sometimes it just feels more convenient to write it and run it.
Yeah.
And that was my rationale. But the longer I've been doing this, I'm realizing that over time this is more work. Because whenever you have to like rerun something,
Yes.
of course you have to redo all of it.
Yeah.
So scripts solve that problem.
Yeah.
But it still doesn't solve the problem of like when you come back to something after a very long time and you want to know what you did or you want to like revisit it, it makes it harder because the computation is gone and you lose some of the context. Whereas putting things in a notebook, I've been trying this and I do think it's better.
Okay.
Like it's partly because to do it, you already have to put more effort into making it in a notebook. So that way you are like covering up some of the loose ends I might not when I'm working with script.
Yeah.
Again, technical details not needed, but basically it has better documentation and it can also do nicer presentation. It looks nicer. So yeah, that's what I've been kind of working on. But the struggle is, this part is okay, but like I also wanted to do slides with it. I think I told you this long ago, I don't know if you remember. And I even told that I'll probably come back to it. I don't know if you remember. At that time I was saying, oh, this is too difficult, the formatting is not working.
Yeah.
So I decided I will learn some HTML, CSS. So I can do the custom styling and make it look exactly how I want it to look.
Okay.
I hate to look at ugly slides, but my slides are always ugly. But the thing is, I can't be bothered to fix it, right? But I feel like if there was an automated way, like I don't have to manually move things around, I can just tell the computer, put it at this meaningful distance from this space, then somehow it becomes easier. So that's part of why this project is make good slides with computers. So that's the challenge, and the experiment will be learn some HTML and some CSS and see how that goes.
Ooh.
Okay, I guess then we segue into the topic. Might be a decent segue,
I think so too.
which is wet lab versus dry lab. I feel like both of us are not, we won't do this topic justice because we are both biased. So let's do it this way. You be the for dry lab person.
Okay.
Because I've been doing this for way too long, I'm kind of bored of it. So I will try my best to make the wet lab case.
Okay. Cool.
Okay, so before we even start, I just wanted to think about this. Wet lab is like a funny definition, no? Like why is it called wet lab versus dry lab?
I was thinking this as well. Like when
Yeah.
you texted me about wet lab, I started. I will tell you actually another story. So this is not really exactly wet lab, but when I met one of my housemates for the first time, this was during like when I went to see the house, and that time I had gone to Chinatown. I came to lab to do some work, and then I went to the house, and I was chatting with them, and then I told her, oh, I went to Chinatown. I just looked around. It was some festival, I think, their New Year's or whatever. And then I went to lab. I did some cell culture, and then I came to see the house. And then this girl, she thought I sold culture.
You sold culture? What does that mean?
Like from Chinatown, I got some culture, and I just sold it in the lab.
What the
I
fuck?
don't know. They just thought about the SELL.
Oh, SELL, cell culture.
Cell culture.
Oh man. Okay. Wow.
Yeah, but I mean to think about it, who thinks about normal cells?
That wouldn't even make
normally?
sense. What does even? Yeah. I
Yeah,
get it.
the
Like,
cell culture. Like now, it's just such a normal word to us. But
Yeah.
what does it really mean?
like the other meaning of the word is not very popular.
Exactly. Anyways, about wet lab, right? I think it's just like just the liquids involved.
Yeah, but that's the thing. But what if you're like doing physics? Like there is no I don't think like material science.
Oh,
and
is
you're
that
working
called
with,
wet lab though?
I guess so, right? No, I thought because
Oh.
dry lab means like computational stuff.
Correct. Oh, I was really just thinking in terms of biology. I never thought about.
Yeah, like
okay,
what-
chemistry, yes, chemistry. This wet lab actually got me, like, not really got me in trouble, but another time I was just going with this new friend, like, new person who does not know about biology, and I told them I do wet lab. And he's just like,
What does that mean? Is this like a sex thing?
exactly. Like, okay, I'm just seeing you right now, but okay, people are getting pranked.
Yeah. Okay. Anyway,
Mm-mm.
and also one thing before we move in, I guess our background is also that we are hybrid, right? We are not purely dry lab, and we are not purely wet lab
Yeah.
anymore, at least.
Yeah.
I guess we both started off as wet lab,
Yes.
and then we transitioned.
Yes.
Are transitioning
Yes.
into dry lab.
Yeah, I guess we can discuss how we got into this as well.
Mm-hmm.
How did you get into dry lab?
For me, it was. I actually hated the idea of code when I
Mhm.
was younger because I was surrounded by people who wanted to become computer scientists, and I very naively. It was everybody, and me being like the hipster guy was just like, oh, I don't want to do what everybody is doing. So to me, it was like that. Like I'm never gonna do that. But I think maybe it started like the first concrete thing was during my rotation with Viju, one of the PIs here. There's a postdoc in the lab who was using R to do some volcano plots, and he's a pretty cool guy. And I think that had a lot to do with it. If he was someone not cool
Yeah.
and using R, I'd be
Yeah.
like, why are you doing this? Just
Yeah.
find some proprietary software. But the fact that he was cool and he was doing it, I was like,
Okay.
oh, this is cool. So I think that's how I started because he wanted me also to try to do that during the rotation. And yeah, I think that's when
That's
when it started, I found like it spoke to me in like so many ways, and I just kept doing it. Like, whoa, this is crazy. Yeah, and the plotting thing has clearly stayed with me.
Yep.
Right, that is one of
Yep.
my core
Yeah.
interests for coding. It's making good, pretty
Plots.
plots.
Right. Nice.
What about you?
For me, I never really thought about bioinformatics. During my masters, during my rotations, etc. I mean, I did not even have a rotation during my PhD. Chose the lab and I went. Yeah, but there was no bioinformatics experience or even like take a little trials like this. But I think the one project that we were discussing about cell-free DNA at one point when I reached, there were different projects being discussed, and like mostly I was obviously planning to do wet lab. But this project required some bioinformatics, and then I just got introduced to some sequencing experiments. So first one was just this RNA sequencing that the lab did, the entire lab did on these cell lines that we had, and then I started playing around with that data because I guess people were doing their own things, but this data was sitting there, so I started playing, started learning, and then I think as things started building up, and then it was like, oh, I have this result, so let me now do this experiment, which will actually build on top of it, give me some new insights. And I guess that's how I started learning, and it was just self-learning as well. There was no bioinformatician in the lab. Yeah, and now definitely I do like it better. I don't really think I am doing dry lab because it's really using packages and only some coding. I really do very minimal coding, but definitely would like to learn more.
Yeah, I mean, it's worth mentioning that. I feel like most people in this space are doing that. How often are people writing their own code?
So this is another question I think for myself as well. So if I want to, do a more bioinformatics job later in life, are the skills that I already have, and of course whatever I will build more in the coming few months, is that enough to get that sort of a job?
Yeah.
What are they really doing?
Yeah, so I mean, I don't know if you've seen like this thing, but there's basically 3 buckets, right? One is like analysis, the second is like building software, and the third is like modeling.
Yeah.
I guess that it also increases in like sort of skill requirement as you pass through them. I think for most labs, if you're somewhere between the first 2 buckets, that's good enough. Like even as a full-time bioinformatician, yes, there will be occasionally some tasks which I guess is not easily accomplished from just having a package. So you have RNA-seq and you can't just go through the default pipeline. But difference from someone who's using just the default packages to someone who can do a little bit extra is not that much, because I think maybe the difference is like just learning some medium language, intermediate language like R or Python, and learn, like, just getting a bit more comfortable with that. Because Bash, I guess, is a bit more limiting. Like, you can do a lot with text files, but I guess once it comes to other things like reshaping data, like there's stuff here. But even here, right, there are packages. At the end of the day, you are using functions that people have like pre-built. You're very rarely hard coding something.
True.
It's like if you're in bucket 1, going halfway to bucket 2 is not so difficult. I think what helped me, I'm also not, of course, not fully into bucket 2, right? Like just slightly beyond bucket 1. What helped me was just, I think the bulk of my whatever I can do is just learning about the tidyverse in R, which is just a set of packages that work with data frames. And that just covers so many of our use cases. Like whatever we want to do, it can be thought of as like a data frame problem. And then you have all these ways to solve it.
Right. Cool.
Yeah. Okay, that was our background.
Yeah.
So that was the main topic. Bioinformatics versus...
Yeah, we just like went all in already.
Yeah, wet lab versus dry lab. Okay. I have one sort of phrasing which I think captures most of why at least I prefer bioinformatics, which is, I think it's very ADHD friendly. Okay, and yes, when I say ADHD, I don't mean like the clinically you have a problem ADHD, go get treatment, but just like a collection of traits that you might have. Yeah, so, and I just think, so for example, let's say you are easily distracted, or you are like interested in many things at once, and as a consequence of this, you don't stay on one track for too long. You start something and then you switch. Right? So this is one. But there's also the flip side of this. Like sometimes you are just very motivated to do this one thing and you just want to start and see it to finish.
Yes.
No matter how long it takes. So both these sort of ends of the spectrum, I think, can be accommodated by wet lab, right? Because you can start an analysis.
Dry lab.
Dry lab. Because you can start an analysis, and then if you don't want to do it, you can freeze it there.
Yeah.
Like you don't lose anything, right? And then you can come back to it later. But with wet lab, obviously that's not possible. I just had this experience, very ambitiously seeded lots of cells for IF. I even put them on the cover slips. Everything is done. Then next day, no motivation, and I just didn't do it that day, and then I have to restart all over. Common problem. But the flip side again is like, so now again today I decided I just want to focus on this one angle, and I just want to, like, this is gripping my attention. I just want to do it till it's done.
Yeah.
Bioinformatics, I have the option.
Yeah.
Wet lab, not possible.
Absolutely.
Right? I have to plan like 2-3 days in advance.
Absolutely.
And I have to trust that I will stay interested in this problem.
Staying interested is one thing. Okay, I will give you all the motivation. I can give you manpower. But even after that, you don't know where like things go wrong. I mean, that with coding and stuff, something went wrong. You can just really come back to it, really find out quickly what's happening.
Okay.
And then it will just take a day maybe. Something like that.
So the quickly thing is another part. So it has like these shorter feedback loops, right? Like you can troubleshoot much more quickly. Yeah, I totally agree. So that's another big part. But just like this thing of like, I want to solve this problem today. I'm okay to sit 24 hours and do it. You can't do it.
Yes.
Like fucking incubation, man.
Yes.
Overnight incubation. It's like, shit.
Agreed, man.
Go home now.
Yeah. Today, I mean, I'm telling you, the day I just came back from my trip, and in 1 day's time, I have like found out results that I want from 2 different data sets, 2 different systems altogether, and which are really useful for my publication. I have that information right now.
Yeah.
I know it's I know it's true for a fact, instead of just repeating it 3 times again and then like getting different results again and again.
True.
It's very satisfying.
Yeah, and then I guess I have one more thing, which is you have the ability to solve problems permanently. So what that means is like if there's a task you have done, and this is a task you think you will do again and again, you can just automate it, right? Write a function, write a big script, and then you don't have to worry about it. But with wet lab, of course, no matter how good you get at the Western blot, you still have to manually do that.
I just thought of Western blot as well. Even if you have done 100 times,
even if
new samples may or may not work.
That's another one. Okay, maybe this is the point where I switch over at least. I'm going to be pro at lab. Although I'm not sure if this is a pro.
Go on,
So,
let's see.
The other flip side of this solving problems permanently is that sometimes repetitive tasks can feel very good, right? Like very meditative. Like this is something you are super used to doing. You don't have to put any brain power, so you just go and you do the thing. So long TC for example, it's very automatic, right? And sometimes that feels kind of good.
But I can do like other 10
Yeah,
tasks
exactly.
in my life,
So
which can be
that's why I didn't want to.
just like fold my laundry or just wash
Focus
my
on that.
dishes.
Yeah, but one tiny thing.
Sure.
Sometimes it feels
I
good.
mean, I shouldn't bash on that so
Yeah.
much.
Okay. Now
Did I have any positive for wet lab.
I have a few. I did my research. This actually you mentioned, I think last time, and I went against it because
I know what it is.
I still feel it, but I think maybe I see it better now also. Also because I was talking to Alexia and she made a similar point. So then I'm like, okay, maybe this is true, which is that results can feel more concrete or like more conclusive.
Yeah.
Do you want to build on that?
Build on that. I mean, I think firstly it's the visualization. For example, immunofluorescence and things like that. You actually just see it there, or say something like a structural model, not modeling, actually looking at a crystal structure or something like that. I just feel that it's something that is not possible with computation. Like results like that or insights like that are not possible to get through computation.
Yeah, fair enough. Like I also don't know exactly what it is because I still feel like a well-designed.
But it is like however well designed it is still limited with this data that I have. But I just feel like there's just more freedom in wet lab, given that you have those equipments and things like that, and then you can just find so many different kind of things.
Yeah, I mean, I don't want to push back on this too much, but like if you change the frame, so for example, when you say the freedom thing, you can also think of it as like you're not stuck with the data you have, right? There's other data sets out there.
Right. But here, I mean, you have the liberty of going and generating this new data by yourself also.
Yeah.
The cell line you have, you ask this question, is this thing affected? You can go and just address it.
Yeah, so I guess here the difference is, so like when I talk about dry lab, I guess I still being this hybrid person, I still usually think of it as like
a collaboration in between,
or even I can do a NGS experiment and then analyze the results.
Sure.
Like
I think I am really thinking about wet lab versus dry lab.
Yeah.
But of course, in the real world, we are collaborating, and that's how things are going on.
Yeah, and because like my work, all the data I analyze, most of it I generated, right? It's ChipSeq, things that I work with. So although like there is like the sample prep doing the whole chip, which is the wet part of it, I tend to think of it as a dry lab experiment. Maybe that's where the bias is coming from.
Fair.
Yeah. So, okay, it can feel more conclusive. And I think the way you said it reminds me of another point that, again, Alexia mentioned, but I agree, is that it's more tangible in the sense that when you're doing the experiments, physically interacting with objects, moving things around, touching things, I guess can feel satisfying. Right? Like
I think so, yeah. Like you're growing these organoids.
Yeah.
You're seeing them grow into like these spheres and things like that, and then you're doing stainings of whatever markers. You can actually see these things grow. Versus now you have these organoids, you sequence them, and then you're like just looking at whatever all the
like so many steps removed. Yeah, I wonder maybe we are doing wet lab in the wrong century, because before I assume it was way more hands-on, right? So I was reading this book, Letters to a Young Scientist. I think I mentioned it.
You have mentioned it.
So he's this guy who works with ants, and his experiments. So he has this great story of how he got into the research. It's just some kid who's like scarily interested in ants, like weirdly interested in ants, just obsessed with ants.
Yeah.
And then he decided he wants to do a PhD on it. And his experiments are like, what happens if I pull out one leg? Or like, what happens if I put this chemical on its head?
Oh man.
He's literally physically holding the ant and like putting drops on it and then letting it and seeing what it does. And there is no boundaries because nobody has tested anything, right?
Yeah.
So the concept of a pheromone doesn't exist. So he's asked his friend to synthesize a bunch of chemicals and then he's putting it on one ant and then seeing what the other ants do to it. And then if you put like some pheromone which comes from a rival ant, the ants like gang up on it and like tear it up into pieces, and he's just watching all this and taking notes.
What a psychopath, man.
But I can see how that's very gratifying. Like it's very physical, right? Like you look
Yeah.
like that.
Mm-hmm.
Yeah, so that part I can agree. I think that would feel more engaged
Yeah.
than like looking at some numbers on a screen.
Exactly. Like even the IHC I have done. So the same thing. It is basically just looking at this histone modification after and before and after treatment. One way I have done it is using chip sequencing. I have the data which I'm really happy about, whatever I'm seeing. But then seeing it on tissues with the staining, and you can see the morphology, you can see it's really high in this sample versus the other sample. It's quite cool.
Fair. Yeah, makes it more visual. Okay, and the last one I had at least is something I think of as a con, but again, maybe with the right, maybe this is just like different personalities of people, right? And depending on that, it can be a pro or a con. All of the things that we said before.
Absolutely.
So this was Alexia's point where it gives you more structure because you have to plan things in advance. It sort of makes you more disciplined. Like you have to stick to it, otherwise it's not going to work, right? So it's giving you some incentives to like plan your day, so that I guess you don't naturally, like I guess for her the concern was if she doesn't plan things up, she might end up doing nothing, and which feels like shit, right?
Of course.
So some structure is like appreciated.
I understand that. Like a lot of the wet lab people that I have known, very well organized, very well structured, and they really follow things through. But again, for me, I think I would like the flexibility of being able to do things again whenever.
Yeah, the reason I don't accept this is like I've told you this in so many different ways, but my solution to this problem of you are not a structured person is to like double down on it. Like do the things whenever you want to do and just trust the process, and eventually things will get done. Like you come back to things you have dropped off. In the long run, I feel it's more efficient. So my strategy has been to not fight against it, but embrace it. But I can see like how that's...
I think I understand that, but for wet lab stuff, because I have been struggling with this as well.
It doesn't work for wet lab.
Yeah.
Yeah, it doesn't work for wet lab.
That's all I was going at.
No,
like for
it
wet lab,
doesn't.
it's not going to work.
It doesn't. It's a,
if you're like planning on doing like embracing that in wet lab, also not a great idea.
Well, it depends on how far you will embrace it, right? So right now, I just seeded some cells and I threw them. I could take that as a loss, or I can just be like that's part of the process.
Yeah,
and
once in a while, it's absolutely okay.
Yeah.
I was just gonna ask, like, do you have any advice for a newbie who is a wet lab person and is trying to get into some bioinformatics? Because I think even if forget about the discussion about wet versus dry lab, this is something like this hybrid is something that's needed for the future, I think. So yeah, if anybody new wants to learn this.
Okay, I guess my advice would be learn Python, probably because it's like quite flexible. Although I myself lean towards R, but I think it's like a wiser step to learn Python. I think Python and Bash together can really get you very, very far, like more than what you would need for like some bioinformatician entry-level job. How there's like tons of resources. I think Code Academy or Data Camp, both of them are quite good, because as opposed to learning from some video series, they give you like a workspace where on the left you have a problem statement, and then you write functional code to solve that problem in the center, and on the right you get your output. So it's like a very get your hands dirty kind of way, and it has like, again, it's because of like this fast feedback loop thing. It just feels very rewarding, and you can very quickly learn a lot. And then you can skip. So the way they do this is like they don't even give you much theory, and most times you don't need the theory, right? You just need to learn how to do things, and that can take you a long way. And honestly, that's how I started, because like that was so fun to do. I didn't require.
Like a game.
Yeah, I wasn't even thinking like, oh, this is good for me for my career. I was simply
just
doing it
having fun.
because that was like interesting, which I think is probably how you should decide if you want to do something anyway. Yeah, I think that's about it. The rest is all logistics.
Yeah.
Okay, so the outro was supposed to be some discovery, right? But what I had was basically the Oppenheimer soundtrack. I don't know if you were following the conversation on the Telegram group, but I was basically asking people for tracks that they listen to when they need like motivation for work.
Hasn't this happened?
This has happened. I do this routinely
Okay.
because I want to update it. So Andy was saying that he likes the Oppenheimer soundtrack, and actually I never paid attention, but then I went back to it and I listened to it. It's very good.
Okay.
Like it does the job and it does it really well.
Nice.
So it has this element of like a seriousness, right? Also you associate it with the movie,
Yes.
so it works like double the magic. So if you have seen the movie and you're looking for some motivational music to listen to while working, Oppenheimer soundtrack.
I think it's the Oppenheimer vibe.
Yeah, I think it's the vibe, what you associate with it.
Yeah.